Great. Thank you, Bruno, and thanks to all of you for tuning in. Can everybody see a little creature moving about on the screen? Okay, great. All right, so very happy to talk to you today about what I see as the future of AI, and I think AI in the coming years and decades is going to look very different from ChatGPT and Claude and Stable Diffusion and Cdance 2 and so on. So what I thought I would do today is show you two candidates, two different kinds of technologies that I think embody what AI might look like in the future. Before I talk about those two technologies, just a couple of housekeeping notes. Unfortunately, I'm going to have to give this presentation in English. I know that many of you on this call, English is not your mother tongue. But I can guarantee you that your English is better than my Spanish. So I will present in English. I don't have much to present. I'm going to go relatively slowly, and I can see the chat. So please don't hesitate if something was unclear to type into chat. I'm happy to pause and go back and re-explain anything. I'd rather talk with you than talk at you. Okay, so let me start by talking about what I hope the future of AI is, and then I'll talk about Xenobots, and if there's time, I'll talk about polycomputers. So what does AI and robotics look like at the moment? This is my best attempt to visualize the current state of AI and robotics. Obviously, the world around us is filled with things other than tokens. So in machine learning and foundation models and large language models, they talk about token prediction. We humans have to boil all the rich complexity out there in the world down into symbols, A, B, C, D, E, or words, or pixels, or whatever it is you consider a token. And then inside the machine, it's relatively empty. There's not a lot of richness going on. There's simply token prediction. Now, arguably, it happens very fast, and it can become very accurate with enough data, as we all know. But if you then look at sort of the same visualization for organisms, things look very different. Every plant and animal on this planet has a rich inner life, much more rich than the inner lives of any of our AIs or robots. What do I mean by a rich inner life? If you zoom in to an organism, you see sensors and motors, you see sense organs, and you see muscle groups. If you zoom in again, you see tissues or organs. You see heart, you see lung, you see intestine, and each one of those organisms is also an agent. It is also capable of sensing its surroundings, processing that input, and acting on its surroundings. If you zoom in again at the cellular level, a cell is arguably one of the most complex sensor motor agents out there. So one way of looking at organisms is they are agents all the way down. And at every level of organization, the whole body, the organ level, the cell level, there are agents that are sensing, quote unquote, thinking and acting and negotiating with their neighbors. If you take the most sophisticated autonomous vehicle, a self-driving car, the car itself has sensors and motors. But if you zoom in on the rubber of the tire, or you zoom in on the steel of the chassis, or you zoom in on the plastic of the front bumper, you no longer see anything that senses, thinks, and acts. You get to inert materials, rubber, plastic, or metal. And those things, by definition, because they are inert, they cannot adapt and react. What that means is that an autonomous vehicle only has one line of defense if something goes wrong. So for example, if a self-driving car receives some sensor values at the level of the whole car, the LiDAR, the cameras, the inertial sensors inside the car, if that incoming sensory percept is far outside of its experience, if it's an out of distribution perception, there's nothing the vehicle can do. There's no backup to figure out what to do in that unfamiliar situation. In the case of an organism, if the organism is surprised, if the organism senses something that is far outside of distribution, it's important to remember that that sensation, whatever unfamiliar situation is happening to the organism, it is flowing down and into the organism and reaching many of the tissues and organs and cells inside the body of the organism. And in that vast collection of cells and organs that are receiving parts of this unfamiliar situation, it is probable that some of them, when they get their local slice of this unfamiliar situation, that little slice is familiar to them. It's unlikely that a surprising event would overwhelm all of the billions and trillions of cells inside your body. Some of your cells are going to know what to do, and they can, because they are not surprised, they can start spreading non-surprise or appropriate reaction back up to the tissue level, the organ level, and back to the whole organism level. And you now have an appropriate response to a surprising situation. So before we talk about any specific technologies, the argument or the thesis that I'm trying to communicate to you is that as we move forward, part of the problems that we're struggling with at the moment with AI and robotics is because they look like this. They are sensor motorically hollow. At the smaller scales, they can't react. So today, I'm going to show you two quote-unquote robots or technologies, or at least smart technologies that do have this rich inner life. And I'm going to show you how that rich inner life prepares them for the richness and surprise that the real world throws at us. So I think in future, what we need to do and what we should do and hopefully will do is create technologies with rich inner lives. And those technologies can use that internal richness to prepare themselves for dealing with external surprise. Okay. That's what I see. That's what I hope for the future of AI. Just to give you, again, more of a picture of what I mean by this rich inner life, here's that inner life at work. What you're watching is a developing salamander, which you have in your corner of the world. We do not have here in Vermont. You just saw cell division. Now you're seeing invagination. So the creation of an inner tube, which becomes the mouth and the anus of the salamander. And you can see all this rich internal dynamics going on inside the developing salamander itself. I want you to think about every individual cell. You can see some of those cells migrating, moving about inside the body of the salamander. I want you to put yourself in the shoes of any one of these cells. I want you to think about the breadth of sensor and motor experiences that all of those cells are getting during development before this salamander ever leaves the egg. You can see that it's still inside the egg here. It would be it's hard to imagine that all of those rich experiences that those cells are acquiring don't help the overall organism, the salamander, adapt to whatever the world is going to throw at it when it eventually leaves the egg, which it's going to do in a few seconds in this video. That's what I mean by a rich inner life. And this is something that we increasingly refer to in my lab as morphological pre-training. You can use the inside of your body to pre-train yourself, to prepare for things outside. Okay. All right, let's talk about the first of the two smart technologies I want to talk about today. The first one is Xenobots, and if you haven't heard about Xenobots before, I'm going to explain what they are. This is a Xenobot that you're looking at here. A Xenobot is about 1 millimeter in diameter, so about the size of a poppy seed. It's very small. We're looking at this particular Xenobot through the microscope. And this Xenobot is made up of about 2,000 frog skin cells that have been taken from a host frog. You can see the individual cells here. Xenobot is Xeno, Xenobot. Xeno is short for Xenopus laevis, which is the Latin name of the frog that donated its cells to make up this Xenobot. Some other things you can notice here. You'll notice these white flashes. These are the white flashes are sudden increases in calcium concentration inside the cell. Cells use a lot of mechanisms to communicate. One of them is calcium. So you're looking at sort of a community of 2,000 cells that seem to be communicating with one another. These cells have been put together in an arrangement that they've never been put in before. Remember that we took these cells from a normally developing frog. When we take those 2,000 cells and smoosh them into this new situation, not only do they not die in this brand new, very surprising situation, but they also inherit or keep a lot of the adaptive ability that the original frog had. For example, in this video, you're going to see a very, very bright flash of white in a few seconds. That big flash of white is Doug Blackiston, our microsurgeon, piercing the Xenobot with a very small pin. So basically hurting or wounding the Xenobot. And you'll notice that the cells nearby almost immediately start to migrate into where the wound occurred. So these cells, although they're in a surprising situation, a group of 2,000 cells, and then they're also injured on top of that, from their perspective, this situation is not surprising enough that they don't know how to do what they're supposed to do, which is move the cells into the wound and heal it. So this is a Xenobot, and what I'm going to show you now is how my group and Mike Levin's group at Tufts University brought in some AI to take this kind of raw material and build robots from them. So instead of building robots from metal and plastics and ceramics, I'm going to show you now how we can build robots from frog skin cells and other cell types. Before I do, one other recent finding from our group that I think is quite interesting and is worth talking about. We reported this in the Communicative and Integrative Biology Journal last year, and that it's this flashing of calcium that you see, it's possibly a signal of how adaptive and how smart this material is. And therefore, it maybe makes sense to make robots out of this smart material, frog cells. So I just showed you a video of a Xenobot, whole bunch of cells, and we can watch over time increases and decreases in calcium concentration. So we don't know what these cells are communicating to each other, but we can sort of see these sort of shouts, which are these spikes in calcium concentration. One thing we can do is to ask whether it's just random noise. Maybe they're not actually communicating. Maybe these cells that make up a Xenobot are just kind of individuals and they're kind of freaking out because they've been put together in this new situation. So in order to answer that question, my postdoc, Thomas Varley, took time series data from each of the cells in a Xenobot, represented by each line in this graph. Increases in the graph are increases in calcium concentration. Drops in the graph are decreases in calcium concentration. And then we can visit each pair of cells, each pair of lines, like the top two, for example, and we can ask how much or how little they covary with one another. So just looking at this top one and the second one, it doesn't look like they covary very much. It kind of looks like they're somewhat independent. We can compare those first two cells, and we can compute the covariance between them, and that gives us a positive number if they covary. It gives us a negative number if they inversely covary, and it gives us a number near zero if those two signals seem to be independent. We can then visit the first and the third cell and again compute covariance, the first and the fourth, and so on. And we can build up a covariance matrix, which you see here, where every red pixel at position ij is reporting that cell I and j seem to be flashing together. They're covarying. Every blue pixel here are pairs of cells that are negatively covarying. Whenever one flashes, the other one doesn't, and vice versa. And the whiter pixels that you see, these are pairs of cells that are relatively independent. If you now take this covariance matrix and you permute columns and you permute rows so that similarly colored pixels are near one another, it turns out that you get something that looks like what you see on the right. You get these blocks of cells or groups of cells within the bot itself that are covarying. There seem to be sort of little communities of cells inside this blob of 2,000 cells. So there seems to be some inherent structure going on inside this smart material that we're going to try and build robots from. Interestingly, if you take a human and you put them in a brain scanner and you scan multiple parts of the brain, different brain regions. Yes. Go ahead. Yeah, go ahead. Oh, it's a great question. No, they're not neighbors. These four groups seem to be spread out throughout the body of the Xenobot. Why that's the case, I don't know. I see there's another question in chat there. Actually, I take that back. I can't see chat. Did someone ask a question in chat? Yep. First of all, for my colleagues at Tufts, it's the easiest thing for them to image. It's still difficult, but it's the easiest of many of the ion species to image. And calcium is one of the primary modes of communication among cells. We could do it with potassium and other communication vehicles as well, but we haven't yet. Just calcium so far. Yeah, good question. Yeah, so I'm sorry, I can't see chat. Feel free to unmute yourself and ask questions as I go. That's fine. Interestingly, again, so if you scan different brain regions and now you're looking at changes in oxygen levels, so basically how hard are different brain regions working, you get a very, very similar picture. You get community structure inside human brains. So Thomas was just interested in how similar this internal structure is in different cells inside a xenobot and different regions inside a brain, and they are remarkably similar. You can conclude from that what you want. The most interesting conclusion is that bunches of frog skin cells are possibly communicating like neurons do. Maybe the xenobot is thinking, maybe it's conscious, who knows? It gets the philosophical implications of this finding become very interesting very quickly, and I'm happy to talk about that at the end. For the purposes of today is when we build robots out of this kind of material, we are not building with inert materials. They have their own ideas about what they want to do, and also their adaptive potential. Okay. So I mentioned that we scrape cells from a host frog and we kind of smoosh them together in a blob. That led us to the idea that maybe instead of us taking cells from a host organism and rearranging them, we can turn over that cellular rearrangement job to an AI. And that's what we did about 7 years ago now. So at that point, our Tufts colleagues told us that they could collect 2 cell types from early frog embryo, frog skin cells, and frog heart cells. Frog heart cells, like your heart cells, spontaneously get bigger and smaller. And if frog heart cells during normal development form up into a frog heart, they spontaneously, they synchronize, and that's good news for the frog. Okay, so we took those 2 cell types and we built them into a biophysical simulator, and you see that simulator running 3 times on the right here. And so you can see that this biophysical simulator is quite approximate. We're approximating cells as cubes. So just a visual reminder that this is a very rough, approximate, and crude. It's a crude approximation of actual frog skin cells and actual frog heart cells. Okay. Once we built this biophysical simulator, we wrapped the simulator in a genetic algorithm, which is a very simple AI search process. The genetic algorithm starts by constructing 100 random xenobots, and you can see the first 3 random xenobots that this genetic algorithm assembled. Basically, we tell the genetic algorithm there are 2 building blocks that you can build from. Skin cells, which are represented by the blue cubes, which are passively being pushed and pulled by the active frog heart cells represented by the red green cubes. It's a little difficult to see in this video, but when a cube turns green, it gets a little bit larger in volume. And when that cube turns red, it gets a little bit smaller in volume. We asked our Tufts colleagues, our biology colleagues, we said, so what happens if the genetic algorithm puts heart cells together in the shape of something other than a frog heart? Will those heart cells synchronize or will they not? And our biology colleagues said, we don't know, nobody's really done this before. So in the biophysical model, we forced the heart cells to not synchronize. You can see the lack of synchrony here. You don't see all the heart cells turning red together and then all the heart cells turning green together. You can see that the spontaneous increase and decrease in volume of the heart cells is deforming the shape of the xenobot, and it's a little hard to see in these videos. But that deformation or that change in shape is causing the xenobot in the bottom right to move a little tiny bit to the right. We also told this genetic algorithm, we told this search process that we want it to discover a way to put skin and heart together to make something that moves to the right as far as possible. So at the beginning, this one at the bottom moves like 1 millimeter further to the right than the middle or the top one does, which basically don't move at all. So they're all doing terrible at what we want them to do, which is move to the right, but one of them is doing slightly less terribly than the other 2. So at this point, the genetic algorithm deletes the top and the middle xenobot and makes randomly modified copies of the surviving xenobot that you see at the bottom. In truth, this genetic algorithm starts by making 100 random xenobots, and I'm just showing you 3 out of those 100. So in essence, what the genetic algorithm does is it assigns a score to each of the 100 xenobots. It deletes the lower scoring or the slower moving bots and makes randomly modified copies of the higher scoring or the faster moving bots. This search process is very simple as I just described it, but it's very computationally inefficient. most randomly modified copies, the offspring Xenobots, do worse than their parents, so they're deleted. And you repeat this process generation after generation. It's very computationally costly, so we had to run this genetic algorithm on our supercomputer here in Vermont. And after about 2 weeks of running on the supercomputer sent us back this video. This was the best combination of skin and heart that the AI came up with. And as you can see, it does indeed move to the right as we wanted it to do. One other observation I want to make here is that remember that the heart cells are desynchronized. So in essence, this is a robot with motors that aren't synchronized. They're kind of randomly doing their own thing. But the skin that's on top is soft and passive, so it's absorbing all the individual forces that are coming up from the heart cells, and that skin is kind of averaging or smoothing out all those random actions to produce one larger force, which pushes down and to the left, causing the robot to move in a relatively straight line to the right. So this is just a reminder that when we ask AI to design things for us, like, for example, a robot, it's able to do things that are very difficult and non-intuitive for human engineers to do. In this case, to make something that moves predictably, moves in a straight line, using unpredictable parts, desynchronized heart cells. That's the end of phase 1 of the experiment. So the AI has basically generated a robot design. We sent this video to our microsurgeon at Tufts, Doug Blackiston, and then Doug got to work looking through the microscope and building a physical version of that simulated Xenobot. What you're watching in this video here are the early stages of this construction process. At this point, Doug has scraped off or has collected some cells from a host frog. And these are the frog heart cells, which is the first layer that he put down. So we're basically looking down through the microscope into a petri dish. That petri dish is filled with room temperature fresh water, which is where this particular frog tends to be most happy. And this large dark circle that you see in the center, this is a small concavity, a small hole in the bottom of the petri dish. And Doug has sort of sprinkled these heart cells into this hole, and the cells are kind of settling onto the bottom of the hole and forming a little bit of a sheet. So this is Doug putting down heart, because the AI said there should be heart cells on the bottom of this Xenobot. After letting these heart cells settle, Doug went back and got some more heart cells and let them settle on top of the first layer of heart, then another layer of heart, then another layer of skin, another layer of skin. And Doug is kind of building up the general cell distribution of this design like a sandwich. So at the end of this process, he basically has a big ball of cells, like I showed you in an earlier video, where the bottom part is heart and the top part is skin. After that process is done, again, through the microscope, Doug is now reaching in with very small microsurgical devices. They're not even devices. They're just very small very small scrapers. You can see him scraping away or removing the unwanted material. At this point, the bot is kind of upside down. You can see these small little stubby legs, which is where the heart is, and there's the skin on top. When Doug finishes this sculpting process, Doug is going to grab the Xenobot with these very small calipers and put it right side up, put the small little legs on the bottom of the petri dish. And when he does, and he now shoots a video from above, again, you can see the real Xenobot moving to the right in more or less the way that the AI predicted that it would. This is known in robotics as the sim to real problem, so trying to transfer something from simulation to reality. And our first paper on this topic, which we published in 2020 in PNAS, was basically a proof of concept that this can be possible. We can actually get an AI to design a robot that's made up of biological cells in simulation, and then they can be manufactured by a human, in this case, our microsurgeon, Doug. A couple other things to note here is you'll notice that the 3D shape of the physical Xenobot is different from the simulated Xenobot shape. And it also moves slightly different. So the transferal from simulation to reality is not perfect, but it's pretty close. So again, we're able to take a raw material and put it together in a way that that raw material has never been put together before, and we get something that's, again, doing a very simple task like locomotion. But to me, back then when we were doing these experiments, this suggested that for certain applications, maybe many applications, we should be trying to build robots from biological rather than technological components. I don't have time to show you this, but it turns out that you can injure this physical Xenobot and it will recover to some degree. This is obviously difficult with physical robots. If you cut off one of the 4 tires of an autonomous vehicle, it's unlikely to behave properly. You make a vehicle or you make a robot out of living materials, and that may not be true. It may be able to regenerate or recover from surprise better than a robot made from technological parts. That still remains to be proved. That's off in the future, but I think this sort of created a new branch of robotics, a new way to think about creating autonomous and safe machines. Okay, that's the end of part 1, and I'm going to spend about 15 minutes on the next technology, and then I'm happy to take questions. But if there's any specific questions or comments about the Xenobots, I'm happy to take them now. Okay. We're going to switch now to, again, a technology that's not a robot, at least not yet. And what I'm going to show you is a material that is able to compute, to do computation in a way that's very, very different from any computers that have existed so far. Again, this technology or this material I'm going to show you is very different from robots. It's different from GPUs, CPUs. It's different from traditional robotics, autonomous vehicles. But like the Xenobots, the material I'm going to show you in a moment has a rich inner life. There's a lot of complicated things going on inside this material that allows it to compute in an interesting way that is very different from the way that traditional computers compute. Okay. I'm gonna introduce you to this material step by step. Let me start with something that I hope for many of you on this call is familiar. The basic building blocks of most computers are Boolean logic gates. Most of those logic gates take 2 inputs and provide take 2 binary inputs and produce 1 binary output. I'm going to start with the NAND gate, or not AND. This particular gate, the NAND gate, in this case, if it takes 2 zeros as input, the green 0 and the blue 0, it should produce a 1 at its output. So 0, 0 goes in as input and a 1 comes out. If instead, for this NAND gate, we supply it with 0 and then 1, it should produce a 1 as output. Third case, if we give it a 1 and a 0 as input, it should provide a 1 as output. And in the fourth case, if we supply 2 1s as input it should produce a 0 as output. This is the NAND gate. It turns out that the NAND gate among all Boolean gates provides universal computation, meaning if you want to build a computer, no matter how complicated that computer is, you can basically build it as a network of NAND gates. So this is why we started with the NAND gate. You're now looking at what's called a granular metamaterial. So let me start with metamaterial. A metamaterial, unlike materials you find in nature, has been designed by us to have interesting properties. You can make metamaterials out of different building blocks. Granular metamaterials, as the name implies, are metamaterials that are made from grains. These could be pieces of sand. They could be pieces of rice. They could be all sorts. It doesn't really matter. The individual grains themselves are inert. They're not smart. They're not like the frog cells I was just showing you. They're inert material. But if you pack them together in a certain way and you design the features of those grains in a certain way, you can get the material as a whole to do interesting things. And what I'm about to show you is a granular metamaterial that's been designed to embody or act as a NAND gate. Okay, let me tell you a little bit more about this video so you can better interpret what's going on here. I want you to think of this material not as a rectangle, but as a rectangle that's been bent so that the two short edges bend around and touch one another. So basically you have an open cylinder. That's what you're looking at. So imagine a cardboard piece of toilet paper, the thing that holds toilet paper, a cardboard cylinder, and built into that cardboard is a whole bunch of these grains that are sliding past one another. So we're building basically a sheet that is closed. It's cylindrical, but it's built from a whole bunch of these moving grains. You can shake it and these little pieces move around inside. Okay. You'll notice that there are light gray and dark gray grains or grains of sand. The light gray ones are very soft, and you'll notice that when they touch one another, they interpenetrate. They push within each other. Oh, sorry, I misspoke. The light gray circles are stiff. They're strong, so you can think of them as sand. The dark gray circles are softer. When the dark gray circles push against other circles, you'll notice that they often interpenetrate or push past one another more. You can think of these as like very soft pieces of rubber. So imagine we put grains of sand and very small bits of rubber together, and we decide where to put the sand grains and where to put the grains of rubber. That's what you're looking at here. Like in the case of the Xenobots that I described before, we haven't decided where to put sand and where to put rubber. The AI has figured out where to put it. That's what you're looking at here. You're looking at an AI-generated granular metamaterial. Also, like in the case of the Xenobots, the AI has figured out how to put sand and rubber together to produce a desirable result. We want this material to act in a certain way, which is when we supply a 1 and 0 as input to the material, it should return a 1 as output. We are going to, in a traditional NAND gate inside of traditional electronics, a 1 is represented by electricity and a 0 is represented by lack of electricity. So electrons are the carriers of information inside traditional computers. Here, instead of electricity being the carrier of information, it is vibration that is the carrier of information. You'll notice that one of the two input ports, which is the green grain, is being vibrated. It's being pushed back and forth. So we're supplying a 1 to the green input port by vibrating the green input port. For the blue input port, we're holding it fixed. We are forcing it to not vibrate. That's how we're supplying a 1 and a 0. So there is energy going in to this material at the green grain and the blue grain, and that is in effect supplying 1 and 0 in the same way that we supply electrons to a NAND gate inside of traditional electronics. Okay, we then observe the output at some other grain. In this case, we've just arbitrarily chosen the grain at the top middle of the material, the one that I've highlighted in red. And you'll notice that this red grain starts to vibrate back and forth as a result of the green vibrating grain. So the AI has designed this material so that at least in the case of 1, 0, the material is correctly embodying or acting like a NAND gate. This is already kind of unfamiliar territory. You can imagine 3 other videos for the other 3 cases in which we vibrate or don't vibrate the green and blue particles, and we want to see in the 0, 0 case that the red particle vibrates, in the 0, 1 case the red particle vibrates, and in the 1, 1 case, the case where we vibrate both the green and red particle, we want the red output particle to not move. We can do that, but that's kind of uninteresting. I'm going to make the life of this material even more interesting by doing the following. I've turned this material now not from a, this is a sorry, let me just back up. This is a computational material. It's performing a computation for us where vibration is the carrier of information. I'm going to take this computational material now and turn it into a polycomputational material. This is where a jump occurs. Just to orient you, again, you notice dark particles and light particles. Dark particles are stiff and lighter particles are softer. You'll notice that there's still a blue and a green particle. You'll notice again that we're supplying this material with 1, 0. We're vibrating the green particle and we're holding the blue particle so that it doesn't vibrate. If you now look at there's a couple of other new things here. There is a purple particle that is always vibrating. We always vibrate it, and this is just us pumping some energy into the material so that it's able to compute. I don't have time to go into the details of the purple particle. You'll just have to trust me for today's discussion that it's necessary. If you look at the if you look at the plots underneath the material, specifically if you look at the green plot, you'll notice that there's actually 2 vibrations. There's a slower one that's occurring at 10 hertz. And then inside that big slow one, within each slow oscillation, you'll notice that there's 2 smaller bumps. So there is also an oscillation occurring at twice the frequency, at 20 hertz. So when I tell you that we are supplying 1, 0 to the green and the blue particle, we're actually supplying the 1, 0 twice. We're supplying 1, 0 at 10 hertz, and we're also supplying 1, 0 at 20 hertz at the same time. If you now observe the red particle, you'll notice that if you look at the video, you can see that the red particle is vibrating. If you look at the red plot at the bottom, you'll notice that that red particle is vibrating at 10 hertz. So it's successfully computing NAND at 10 hertz. And you'll notice that the red particle is also vibrating at 20 hertz. So it is also acting like a NAND gate at 20 hertz. So we call this a polycomputational material because there's one material, but inside that material, there are two NAND gates that are operating at the same time in the same place, in the same material, but at different locations on the frequency spectrum. There's one NAND gate at 10 hertz, and there's another NAND gate at 20 hertz. No electronics, no computers on the planet at the moment, with the exception of quantum computers, can perform two computations in the same place at the same time. So again, the theme of my lecture today is this idea of creating materials or technologies that have rich inner lives. Here's a material that's inert, it's not biological, it's purely technological, but it has a very rich inner life. It's embodying two computations in the same place at the same time. So just to convince you that this material is successfully acting like a NAND, let's have a look at exactly the same material. In this case, we're vibrating both the green input particle and the blue input particle. So we're supplying 1, 1 as input, and we're supplying 1, 1 as input at 10 hertz and again at 20 hertz. And you can see that by looking at the graph underneath the video, and you'll see the green line has oscillations at 10 and 20 hertz. And the the blue plot also has oscillations at 10 and 20 hertz. And now I want you to look at the red line. You'll notice that the red particle is vibrating, but much less than it was before. So basically, the red particle is being buffered. It's being damped down to oscillate as little as possible. And it's below a certain amplitude. So in effect, that red particle is successfully outputting a 0 at 10 hertz and again a 0 at 20 hertz. So just to finish up here, I'll invite you to, there's a lot going on this slide. I'll invite you to look at just the 4 red lines. You'll notice in the rightmost red line, there's very little oscillation. And in the left 3 lines, there's much more oscillation. So what you're looking at, again, is one material. You're looking at this one material under 4 different conditions, the 4 different input cases for a NAND gate. And in all 4 cases, it's acting like a NAND gate twice at 2 different frequencies. I know most of you are interested in robotics and evolutionary robotics in particular. This is not a robot and it's not an evolutionary robot, but it is a smart material. And we did evolve this distribution of grain stiffnesses to get this material to act as a logic gate. I want you to think of this as a physical material. Imagine we were to take this physical material and actually put it on a flat surface. As it's vibrating, it would start to move. You'd start to get some motion. So we're now working in my lab at turning this polycomputational material into a polycomputational robot. It's a robot that moves through vibration and quote unquote thinks using vibration. One of the biggest struggles in AI and robotics is what's called the Cartesian dualism. Here in the West, we tend to talk about brains and bodies as separate things, or behavior and thinking as different things. I want you to imagine a robot made from this material where thinking and acting are all resulting from vibrations that are occurring at different frequencies at the same time. In such a robot, thought and action become conflated. They become one and the same thing. If you were to point at that material, it would be impossible to say that part of the material is thinking and that other part of the material is acting. They would be one and the same thing. That's my hope for the future. I've talked a lot, so let me just finish up. Here's my visualization of in the future, hopefully we can do 3 gates at the same time in the same place. let me finish up by thanking all of the people that did all of the actual work that I talked about here. All of the faces on the left here, these are members of my lab, and many of my lab members worked on the projects I described today. All of the faces on the right are my colleagues that helped with this work at Yale University, City University of New York, Tufts University, Indiana University, Tufts, Arizona State, and Harvard. And thank you to the National Science Foundation, DARPA, and my home institution here for financially supporting this work. I will stop there, and hopefully we have a few minutes for questions, if there are any. Thanks very much. Yeah, that's a really great question. One of my PhD students here, Piper Welch, if you go to Google Scholar and put in Bongard Welch, you should find her paper. So she looked at the problem you're describing, which is logical independence. So could you supply 1 0 at 10 hertz and 1 1 at 20 hertz, and it still successfully acts as a NAND gate? The answer is yes, it's possible, but it's hard. Hard in the sense that you need to give the genetic algorithm, which is designing this material, you need to give it much more computation, because during the evolutionary process, you have to give it, different permutations and combinations of different input cases at different frequencies, so that it learns how to quote unquote pull apart those virtual gates that are operating at different points on the frequency spectrum. It's possible. Again, we've only done it with simple Boolean gates and only 2 at the same time. We haven't looked at triplets or quadruplets yet, so we don't really have a sense of how or whether this scales, but it is possible. It's a great question. Yeah, completely unknown. We just arbitrarily chose that number of grains. We haven't expanded the genetic algorithm to be able to increase the size or the density or even the packing of the grains. You saw obviously a regular packing there. You could imagine grains of different sizes that are packed in irregular patterns. We haven't looked at that yet, but that would definitely be a good next step for sure. Yeah, both really good questions. Let me answer them in reverse. So yes, it's probably going to be very difficult when we chain them together. So the other thing that I kind of, skimmed over is the readout. So in order to interpret the output, we need to do a Fourier transform to know the power at the frequency we're interested in. So there is still a lot of external computation that we're doing, using traditional electronics. So that's still a limitation. And if we were to chain together multiple gates and try and build up, a half adder or a multiplexer, as we start to scale up, it gets very difficult very quickly. One of the things that I am very keen to try is, if we chain together multiple gates, do those gates need to be at different places? Do they need to be at different physical locations in a material? Because that's how we would think to do it. But polycomputation says you might be able to have 2 different gates that communicate with one another in series in the same place. They could no longer be at the same time, but they could be same place, same frequency, different time. Now, is that more difficult or more simple for a genetic algorithm or gradient descent to design into a material? No idea. But we're interested in these new approaches to computational density, because otherwise, what's the point? We have fantastically computationally dense materials already in silicon. Let's see if we can try and pack computation into a material in a different way. But yes, at the moment, it's much more complicated than using electrons to communicate information. Absolutely. Back to your question about the Xenobots. My postdoc, Thomas Varley, who worked on this, he did a very good job trying to comb out any of the other confounds. So I should say, again, I simplified when I said he compared Xenobots to brains. What he actually did was to take the Xenobot time series data that I showed you and to do what's called a permuted null in time. So basically, frame shift each individual calcium reading from each cell. And when you do that, and I wish I had kept the slide for this, you get basically a mostly white. So you lose all of that community structure. You preserve all the other first order statistics of the community structure, but of the calcium. So it's not just because there's a whole lot of cells. He controlled for that appropriately. So when I overinterpret his results and say the Xenobots are like the brains, what I should have said is Xenobots are like brains in the sense that when you take Xenobot permuted nulls and brain data permuted nulls, they both lose their community structure. Yeah, great question. So again, let me answer them in reverse. So you're right. So again, when I say there are two gates in the same place at the same time, what I mean is that you can observe two gates in the same place at the same time over a sufficiently long time interval that the Fourier transform will give you an answer. So it's not an instantaneous gate. But I would argue, again, in traditional electronics, you need to wait for electrons to move through a gate in order to make your determination for a 1 or a 0. Traditional electronics is still way faster than us. So you can probably guess where this is going. As I showed you, at least in simulation, these gates at 10 and 20 hertz, we'd like to transpose those gates to higher and higher frequencies. So you would require a narrower time interval within which to make a determination of the output at the different frequencies. And how much of a separation between the different frequencies are needed in order to do that. So very good questions. Very good question. And then your first question was, I'm so sorry, I forgot it. Can you ask it again? Oh, yeah, the thresholding. Yep. So the way that we did this is when we ran the genetic algorithm that was designing the material, the fitness function said whenever the output should be 1, maximize the amplitude at that frequency, and whenever the output should be 0, minimize the amplitude at that frequency. So you can think of the evolutionary pressure in the genetic algorithm is trying to push down the 0 output cases and is trying to push up the signal at the 1 output. And then what we get is there's a band, and you can pick that threshold after the material has been designed anywhere within that interval. So it's kind of the threshold is kind of arbitrary. We just want to see a large separation between 0 and 1. Yeah, a very good question. So so in the case of polycomputation, as I showed you, as you can probably imagine, there's a gazillion ways you can put soft and hard grains together in which you don't get what you want. It doesn't work. This is true of all technologies, right? It's a needle in the haystack problem. The genetic algorithm is searching for just that arrangement of grains that embodies the computation that you want. We've looked a little at the robustness. So again, if the stiffnesses are disrupted a little bit, or one of the grains pops out of the material and you have a gap, how gracefully does the computation start to corrode as a function of the amount of perturbation? We haven't done enough investigation to know whether that degradation in computation is better or worse than degradation in traditional electronics. I don't know. We will see. It's a good question. I don't know. Nothing is stopping us other than we just haven't tried it yet, And again, it comes back to like the signal to noise ratio. How much can you pack in, given the constraints, the number of particles that you have, and that it's wrapped in this cylinder. I don't know. We don't know what the upper bound of computational density for a given material with a certain number of grains. It's unknown at the moment. To me, that's kind of the most exciting thing. What is the upper bound? Unknown. Yep. Maybe two more questions. They're great questions. I'd love to keep going, but there are other demands on my time. That's a fantastic question. So, as you mentioned, we did what is usually done in evolutionary robotics, which is maximize displacement. I have no idea how much energy the Xenobots expend, as they move. So, but this has been studied, at the small scale, at the millimeter scale. So there are organisms, that walk along the bottom of freshwater ponds, and they're about a millimeter in size. And the energetics of those species are known. So it would be awesome to see, first of all, step one, just as we did, how energy efficient are the Xenobots at walking compared to other species in the same niche of the same size. And then, as you said, we could incorporate an energy efficiency metric into the fitness function. So, for example, not surprisingly, the more heart cells that are in the Xenobot, the more energy that's expended. Heart uses a lot of energy for obvious reasons. So I think it would be amazing to see if we can make very small biological robots that move in an energy efficient manner. Yeah. I mean, these questions have been studied in robotics for decades, and we can just ask exactly the same questions and try it, but in this new robot material. And do we get similar answers or different answers? Yeah. Great point. To be seen, to be determined. Yeah, it's a fantastic question, and it's beyond my area of expertise. I am a computer scientist, not a biologist. What I have learned from my biology colleagues is in some of these Xenobots, the cells that make up the Xenobot came from the same frog embryo, and they came from the same place in the embryo. So some of the spatial structure for that piece of the Xenobot is preserved from the host organism. That being said, Xenobots or developing frog skin tissue exhibits cell migration, meaning the cells are actually moving inside that material. You might have been able to see some of it actually in the video that I showed. So they don't necessarily stay where we put them. So it's possible that they're migrating to try and be being close to quote unquote similar cells that exhibit calcium dynamics similar to themselves. This is also complicated by the fact that in some Xenobots, a single Xenobot contains cells from different frogs, or at least different embryos from the same mother frog. So it gets very complicated very quickly. But we definitely could look at, calcium covariance and spatial proximity. And sometimes there's a relationship and sometimes there's not. And teasing apart how or whether that's a function of the different donors to the same Xenobot, or whether whether the cells are actively trying to do that on their own, No, I think no one knows at the moment, but they would be absolutely fascinating topics to investigate. It's a great point. We'll add it to our very long to-do list. Thanks for that question. Oh, okay. One more? Okay. Can you repeat your future expectations? I see two in chat. I see one from Fatima and one from Eric. Fatima, okay. Fatima says, if polycomputation allows a biological material to have multiple layers of meaning running on the same structure, at what point in the complexity of those overlapping, does it stop being an AI designed machine and start to, okay, and when does it start to emerge as a self? Yeah, great question. When does agency start to creep in? I would say that I'm a little bit of a skeptic when it comes to agency. I love Daniel Dennett's idea of the intentional stance. So agency is not in us or in a Xenobot or in a polycomputer. It's in how you observe something else. So to answer your question, I think it's going to get really interesting if we wanted to describe the behavior of a Xenobot, we can describe its behavior more accurately and more succinctly when we take the intentional stance. So if we say something like, the Xenobot now is interested in this, and it will delay its own gratification. You can see it delaying its own gratification by doing X. If it gets easier to describe a Xenobot or a polycomputer using the intentional stance, that's going to be really interesting. I would say at the moment they're still so simple that, talking about the material wanting to be a NAND gate, sounds silly. It doesn't make sense, and it doesn't have much predictive power. But I don't see any reason why, as we start, or the AI starts to design more complex materials, I'll bet you we're going to get to the point where it makes sense to talk about them as having agency. I don't see why it wouldn't happen. But again, we'll see. That's a good question. I would love that. Thank you so much. And these were really great questions, and my apologies that I couldn't get to all of them.